The overall goal of this set of consecutive methods is to visualize and monitor vitality parameters and DNA repair in reviving Bacillus subtilis spores after low-pressure plasma treatment using time-resolved confocal fluorescence microscopy and scanning electron microscopy.
Our method can help answer key questions in the field of microbial inactivation and sterilization. It helps us to analyze the detrimental effect of low-pressure plasma treatment on biological indicators such as Bacillus subtilis spores.
The main advantage of our technique is that we combine several methods: CFU determination, scanning electron microscopy, and monitoring the DNA repair using time-resolved fluorescence microscopy in order to verify the detrimental effects of low-pressure plasma treatment.
To carry out spore production, transfer a 5 milliliter overnight culture of the respective B. subtilis strain, supplemented with appropriate antibiotics, to 200 milliliters of double-strength liquid Schaefer sporulation medium, and cultivate it with vigorous aeration at 37 degrees Celsius for at least 72 hours or longer, until greater than 95% of the culture has been sporulated. Harvest spores in 15 milliliter tubes by centrifugation at 3000 G for 15 minutes, and purify the samples using sterile distilled H2O in repeated washing steps. Check for purity and germination status by phase contrast microscopy, and ensure that spore suspensions consist of greater than 99% of phase-bright spores and are free of vegetative cells, germinated spores, and cell debris. Otherwise, further microscopy experiments can be disturbed. Determinate the spore titer by plating 50 microliters of 10-fold